Welcome to our channel. Today we journey deep into the Amazon's past, not through ruins, but through the very DNA of its people. This is the story of millennia of survival, adaptation, and resilience, a history of an entire continent written in the human genome. Let's begin. Amazon's DNA, a story of indigenous resilience. Part 1, the first footprints, charting the waves of arrival. The story of the Amazon's first peoples, a narrative etched not in stone, but in the polymorphic sequences of their descendants' DNA, begins in the shadow of the last great ice age, a time when humanity was poised to enter the final continental frontier. The prevailing theory, long held and supported by a wealth of archaeological evidence from North America, posits a single, major migration event across the Bering Land Bridge, a vast, now submerged subcontinent that connected Siberia and Alaska. These Paleo-Indian pioneers, carrying a distinct genetic signature, are believed to have journeyed southward through an ice-free corridor that opened up between the Laurentide and Cordilleran ice sheets, or perhaps leapfrogged down the Pacific coastline in watercraft, a route that would explain the rapid dispersal of humans throughout the Americas. For decades, this Clovis first model, named for the distinctive fluted projectile points found across North America, dominated the discourse, suggesting a relatively late arrival, around 13,500 years ago, followed by a swift peopling of both continents. The Amazon, in this scenario, was one of the last places to be settled, a formidable green barrier entered only after humans had adapted to the open plains and coastal regions. However, the genetic tapestry of modern Amazonian peoples, when read with the sophisticated tools of molecular genomics, tells a far more complex and ancient story, one of multiple streams of ancestry and deeper timelines that challenge the single migration paradigm. Genetic markers found in some Amazonian groups, particularly those speaking Gu and Tupi languages, hint at a more ancient lineage, one that is genetically distinct from the typical ancestral Native American profile. This lineage, sometimes referred to as belonging to Population Y, shares a surprising genetic affinity with modern-day Australasian peoples, the Onge of the Andaman Islands, Papuans, and Indigenous Australians. This discovery, first proposed based on cranial morphology and later bolstered by genomic data, sent shockwaves through the anthropological community, suggesting a ghost population that entered the Americas in an earlier, separate migration wave, perhaps tens of thousands of years ago. This wave, if it existed, appears to have been largely overwritten by the later, more successful migration of the first Americans, but its faint signal persists, a whisper in the blood of isolated Amazonian communities. It raises the tantalizing possibility of a Pleistocene Americas that was more diverse than ever imagined, a continent inhabited by multiple founding groups who arrived via different routes and at different times. The genetic evidence compels a re-evaluation of archaeological sites once dismissed as too old or anomalous, such as Pedra Ferreira in Brazil, with its controversial dating stretching back over 30,000 years, while the full story of Population Y remains elusive. Its genetic echo in the Amazon serves as a profound testament to the deep antiquity of human presence in the rainforest. Furthermore, the genetic diversification within the main wave of Native American ancestors also suggests a more nuanced arrival. Rather than a single, monolithic group, it appears there were at least two distinct pulses of migration from Beringia, which paused and diversified before continuing their journey south. This Beringian standstill model, supported by mitochondrial DNA analysis, proposes that the ancestors of Native Americans spent thousands of years isolated in the land bridge region, during which time unique genetic mutations accumulated. Once the ice sheets retreated, these now distinct groups expanded rapidly into the Americas, with one wave branching off to populate North America and another heading south, eventually reaching the Amazon basin. The genetic signatures of these distinct southward expansions can be traced today, mapping onto the vast geographical and linguistic diversity of the continent. The Amazon, therefore, was not a late inhabited afterthought, but a primary destination for some of the earliest peoples to enter South America. Their DNA reveals a complex branching pattern, A series of founder effects and population splits that occurred as small bands navigated the immense network of rivers and dense forests, adapting to its myriad microecologies. These first Amazonians were not merely surviving in a hostile environment, they were actively shaping it, and in turn, being shaped by it, a dynamic interplay that would lay the genetic groundwork for millennia of resilience. Their journey, a saga of immense courage and adaptation, is a foundational chapter in the human story, a testament to the relentless drive to explore and settle every corner of the planet, with the Amazon representing one of the most challenging and ultimately rewarding landscapes they would ever encounter. The faint, ancient signals of their arrival, carried as single nucleotide polymorphisms and haplogroups, are the first verses in an epic poem of survival, the unseen DNA that underpins the enduring spirit of the Amazon's indigenous peoples. Part 2. Genes of the Forest, Foragers of the Archaic Period. Long after the first footprints had been imprinted on the Amazonian soil, a new chapter unfolded, one defined not by epic migrations, but by an intimate and profound process of settling in. This was the Archaic Period, a vast expanse of time, roughly from 10,000 to 4,000 years ago, during which the descendants of the first pioneers transitioned from highly mobile big-game hunters to specialized forest foragers, their genetic makeup being subtly and continuously sculpted by the unique pressures and boundless opportunities of the world's largest tropical rainforest. This era is often misunderstood as a time of cultural stasis, a long, uneventful prelude to the rise of agriculture and complex societies, but the genetic record reveals a period of intense innovation, adaptation, and demographic change. The small, scattered bands of early Amazonians began a deep dive into the ecological intricacies of their new home. Their survival no longer depended on tracking megafauna, which had largely vanished with a changing post-Pleistocene climate, but on mastering the forest itself, its bewildering diversity of plants, its elusive and often small-bodied fauna, and the cyclical rhythms of its mighty rivers. This process of adaptation is legible in their genes. Natural selection would have favored traits suited to a forest environment, such as enhanced immune responses to a host of new tropical pathogens, from parasites like leishmania and trypanosoma to a universe of unknown viruses and bacteria. Variants in immune-related genes, such as the human leukocyte antigen, HLA, system, likely underwent selective sweeps, as individuals with more effective defenses against local diseases were more likely to survive and pass on their DNA. This invisible arms race, fought at the molecular level, was a critical component of becoming truly Amazonian. Beyond immunity, metabolic adaptations were also crucial. The diet of these foragers was incredibly diverse, a broad spectrum revolution that involved exploiting everything from fish, turtles, and caimans in the rivers to monkeys, peccaries, and birds in the canopy, supplemented by a vast botanical library of fruits, nuts, tubers, and medicinal plants. Genes involved in processing specific nutrients, detoxifying plant-based compounds, and managing energy expenditure in a hot, humid environment would have been under selective pressure. The ability to thrive on a diet rich in fiber and sometimes scarce in fats and proteins was a key advantage, a genetic legacy that can still be observed in the metabolic health profiles of their descendants. The social structure of these foraging groups, typically small, mobile, and organized around kinship, also had profound genetic consequences. Living in relative isolation within a vast forested landscape, these bands would have experienced significant genetic drift, the random fluctuation of gene frequencies from one generation to the next. A rare mutation appearing in one small group could, by pure chance, become common in their descendants, leading to the emergence of unique genetic profiles in different regions of the Amazon. This process, repeated over thousands of years across countless small populations, was a powerful engine of genetic diversification, creating a mosaic of distinct lineages that mirrored the basin's ecological and linguistic diversity. Yet, these groups were not entirely isolated. The rivers served as arteries of connection, facilitating not only the exchange of goods and ideas, but also of genes. Intermittent contact between neighboring bands, whether for trade, ritual, or marriage alliances, would have created a complex network of gene flow, preventing complete genetic divergence and weaving a loose but resilient genetic fabric across the basin. This balance between drift and flow is a hallmark of the Amazonian genetic landscape. It explains how distinct ethnic and linguistic groups could maintain their unique identities while still being part of a larger, interconnected human ecosystem. Archaeological evidence from this period, such as the shell mounds, sambakis, found along coastal areas and major rivers, points to populations that were becoming increasingly sedentary, tethered to resource-rich locations. These sites, rich in the remains of fish, mollusks, and other aquatic life, represent some of the earliest examples of intensive resource management in the Americas. The genetic profiles of people associated with these sites would likely show the initial stages of population growth and reduced mobility, a prelude to the more settled agricultural lifestyles that would follow. This was a world without pottery, without large-scale agriculture, yet it was a world of immense complexity and ingenuity, one in which the foundations of Amazonian societies were laid. It was during the Archaic that the deep, encyclopedic knowledge of the forest, the traditional ecological knowledge, TK, that is so vital to indigenous identity today, was first compiled through millennia of trial and error. Every plant with medicinal properties, every edible fruit, every animal behavior was observed, learned, and passed down, a cultural inheritance that ran parallel to the genetic one. This profound co-evolutionary relationship between people and their environment, where culture guided the adaptation of the genome, and the genome enabled the flourishing of culture, is the enduring legacy of the forest foragers. Their DNA is a living library, a record of this deep-time experiment in human resilience, demonstrating how to build a sustainable and enduring civilization not by dominating nature, but by becoming an integral part of it. Part 3. The Terra Preta Makers, Engineering a New Eden. The genetic and cultural landscape of the Amazon underwent a monumental transformation with the advent of a revolutionary technology, one that was not forged in metal but cultivated in the very soil. This was the era of the Terra Preta Indio, or Amazonian Dark Earths, a period beginning around 4,000 years ago that saw the rise of large, sedentary populations who were not merely living in the forest, but actively and intentionally engineering it to their needs. The discovery of these vast tracts of anomalously fertile, charcoal-rich soils scattered throughout the Amazon basin shattered the long-held archaeological dogma of the region as a counterfeit paradise, a pristine wilderness incapable of supporting complex societies. Instead, Terra Preta revealed a history of sophisticated landscape domestication and large-scale agriculture that sustained populations far larger and more complex than previously imagined. This agricultural revolution had profound and lasting consequences for the genetic makeup of Amazonian peoples. The shift from a mobile forager lifestyle to a settled agricultural one triggered a significant demographic expansion. Farming communities, centered around the cultivation of staple crops like manioc, maize, and sweet potatoes, were able to produce food surpluses, supporting higher population densities and leading to a veritable population boom. This expansion is clearly visible in the genetic record as a signature of population growth, a decrease in genetic drift, and an increase in overall genetic diversity. As villages grew into towns and towns into regional polities, the patterns of gene flow shifted dramatically. While the old riverine networks of exchange continued, the social gravity of these new, populous centers began to pull people in, creating larger, more centralized gene pools. The genetic landscape began to reorganize itself around these hubs of civilization. The DNA of modern indigenous groups often reflects the geographic location of these ancient population centers, with core areas of high genetic diversity surrounded by a periphery of related, but less diverse, groups. The creation of Terra Preta was a multi-generational project, a form of ecosystem engineering that involved adding a specific recipe of charcoal, pottery shards, animal bones, and other organic waste to the typically poor, acidic Amazonian soils. This process sequestered carbon, increased nutrient retention, and fostered a rich microbial community, creating a self-perpetuating fertility that persists to this day. This intimate, hands-on manipulation of the environment meant that human and landscape became deeply intertwined. The people who made the dark earths were also made by them. Their diet became more reliant on the crops they cultivated, which would have exerted new, selective pressures on their metabolism. Genes related to starch digestion, such as amylase gene copy number, may have been selected for in populations that adopted maize or manioc as a primary staple. Furthermore, living in larger, more crowded settlements created a new set of public health challenges. Increased proximity between people and the accumulation of waste would have facilitated the spread of infectious diseases. This urban penalty would have driven the selection for more robust immune defenses, continuing the genetic arms race against pathogens that had begun in the archaic, but now in a new, more intense theater. The social and political organization that was necessary to create and maintain these large-scale agricultural landscapes also left its mark on the genome. These were likely complex, hierarchical societies, with evidence of causeways, moats, and plazas suggesting a high degree of social stratification and coordinated labor. Kinship systems and marriage rules, which are central to the social fabric of any society, would have directly shaped patterns of genetic inheritance. Patrilocality, where wives move to their husband's community, or matrilocality, where husbands move to their wife's community, would produce distinct patterns in Y chromosome and mitochondrial DNA, respectively. These social structures, invisible in the archaeological record, can be inferred from the genetic relationships between contemporary communities. The legacy of the terra preta makers is not just in the soil, but in the very blood of the Amazon's indigenous peoples. They were the architects of a domesticated landscape, a vast, productive garden that sustained a thriving civilization for millennia. Their success demonstrates a form of intensive agriculture that was not destructive, but regenerative, a model of sustainability that modern science is only now beginning to understand. The genetic diversity that flourished during this period became the wellspring of Amazonian resilience, a deep reservoir of human variation that would be crucial for surviving. The cataclysmic changes to come, the DNA of their descendants, is a testament to this golden age, a time when humans were not just inhabitants of the Amazon, but its co-creators, a people who learned to build a paradise not by finding it, but by growing it from the ground up, leaving behind a legacy of engineered soils and an equally engineered and resilient human genome. Part 4. A River of Words. The Great Linguistic Migrations. The genetic map of the Amazon is inextricably woven with another, equally complex map, the map of its languages. The distribution of the region's major linguistic families, Tupi, Arawak, Carib, and Gu, among others, is not a random patchwork, but the result of great waves of migration, demographic expansions that reshaped the human landscape of the basin over the last few thousand years. These expansions, driven by a combination of technological innovation, agricultural prowess, and perhaps new ideologies, were powerful engines of genetic admixture and replacement, a story told in the shared vocabularies of disparate peoples and the corresponding shared segments of their DNA. The correlation between genes and language, a field known as archaeolinguistics, provides a powerful lens through which to view the history of the Amazon. One of the most significant of these events was the Tupian expansion, originating in the southern Amazon, likely in what is now the Brazilian state of Rondônia. Around 5,000 years ago, speakers of Proto-Tupi began a relentless and remarkably successful migration. They were skilled agriculturalists, possessing a productive toolkit of crops, including manioc and maize, and a distinctive ceramic technology. They traveled primarily along the major rivers, their canoes serving as vectors for the transmission of their culture, their language, and their genes. As they moved into new territories, they encountered and interacted with the diverse groups of foragers and early horticulturalists who already lived there. The genetic consequences of these encounters were varied. In some cases, the arrival of the Tupi speakers led to a process of demographic replacement, where the incoming farmers, with their ability to support larger populations, overwhelmed the local groups. In these instances, the local Y chromosome and mitochondrial DNA lineages were largely replaced by those of the Tupian migrants. However, in other cases, The interaction was one of admixture. Local men might have been incorporated into Tupian society, or more commonly, local women were taken as wives by the incoming men, a process known as sex-biased admixture. This often results in a genetic signature where the Y chromosome, paternal, lineages are predominantly of Tupian origin, while the mitochondrial DNA, maternal, lineages are a mix of Tupian and local pre-existing lineages. This pattern tells a story of cultural and genetic assimilation where the Tupian language and farming practices were adopted by a genetically mixed population. A similar, though perhaps earlier and more widespread, story can be told of the Arawak expansion. Originating in the central Amazon, Arawak speakers spread over an enormous area, reaching as far as the Caribbean islands, the Taino, the Andean foothills, and the southern cone. Their expansion was less a single directional migration and more a complex, multi-directional diffusion of people and culture, often associated with sophisticated agricultural techniques and a distinctive social and political organization centered on hereditary chiefs. The genetic legacy of the Arawaks is one of profound interconnection. Their DNA links peoples across vast and ecologically diverse regions, revealing a history of long-distance interaction and exchange. Studying the Arawak diaspora through genetics allows researchers to reconstruct ancient alliance networks and trade routes, showing how a shared linguistic and cultural heritage facilitated gene flow across the continent. The Carib expansion, originating in the Guiana Shield region, presents another chapter in this linguistic and genetic saga, often portrayed in early European accounts as fearsome warriors. The Caribs were also skilled navigators and traders who established a powerful presence along the northern and eastern coasts of South America and into the Lesser Antilles. Their migrations also resulted in a complex pattern of genetic admixture as they interacted with both Arawak and other local groups. The Gus-speaking peoples of the central Brazilian plateau represent a different kind of story, one of more localized radiation and long-term stability, a deep time presence in the savanna-like Cerrado environment that contrasts with the riverine expansions of the Tupi and Arawak. By comparing the genetic profiles of peoples from these different language families, a detailed history of population movement and interaction can be reconstructed. Shared genetic markers can confirm linguistic hypotheses about the shared origin of two distant groups, while genetic differences can indicate where and when they diverged. The timing of these population splits, estimated using a molecular clock based on the rate of genetic mutation, can be correlated with archaeological evidence for the spread of agriculture or new pottery styles. This powerful synthesis of genetics, linguistics, and archaeology allows us to write a history of the Amazon that was previously invisible, a history that is not about empires and monuments, but about the dynamic movements of peoples, the spread of ideas, and the creation of new, admixed societies. It is a history that reveals the Amazon not as a static mosaic of isolated tribes, but as a dynamic and interconnected world, constantly being reshaped by the currents of migration, with language as the vessel and genes as the cargo. Part 5. Networks of Blood and Goods, Pre-Columbian Trade. The Amazon, often envisioned as a trackless wilderness isolating its inhabitants, was in reality crisscrossed by a complex and ancient network of trade routes that bound its diverse peoples together in a web of social, economic, and genetic interdependence. These networks, which flourished for millennia before the arrival of Europeans, were the arteries of pre-Columbian Amazonian society, facilitating the flow of goods, ideas, technologies, and, crucially, genes across vast distances. The story of this interconnected world is being rediscovered not only through the patient work of archaeologists, who unearth trade goods far from their origin, but also through the analysis of DNA, which reveals the ghostly footprints of ancient travelers and the genetic legacies of long-distance alliances. The great rivers of the Amazon basin, the Amazon itself, the Negro, the Madeira, the Xingu, were the superhighways of this ancient world. In large, sophisticated dugout canoes, traders, and Travelers could cover enormous distances, connecting communities in the Andean foothills with those on the Atlantic coast. The goods they carried were a testament to the specialized production of different regions. From the west came coveted stone axes, made from hard, fine-grained rock found only in the upper reaches of certain rivers. From the lowlands came intricately woven hammocks, powerful hallucinogenic snuffs, and the brilliantly colored feathers of macaws and toucans, which were essential for ceremonial regalia. Certain communities specialized in the production of high-quality pottery, or the potent curare poison used for blowgun darts. This intricate system of exchange did more than just distribute resources. It created a shared material culture and fostered a sense of a larger, interconnected Amazonian world. Genetically, these trade networks had a profound impact on the structure of human populations. They acted as powerful conduits for gene flow, counteracting the isolating effects of distance and genetic drift. A trader from a community on the Rio Negro might travel to a festival or trade fair hundreds of miles away, and there form a temporary or permanent union with a local woman, introducing new mitochondrial DNA lineages into that community. Similarly, formal marriage alliances were a common way to cement trade partnerships between powerful chiefs or entire communities. These alliances, which often involved the exchange of high-status women, were explicitly designed to create kinship bonds, to turn strangers into family, and in doing so, they directly shuffled the genetic deck, weaving together the DNA of distant groups. The genetic evidence for these networks can be seen in the non-random distribution of certain genetic markers. For example, a Y chromosome or mtDNA haplogroup that is common in one region might appear as a small but significant outlier in a distant population known through archaeology to have been a trading partner. By mapping the geographic distribution of these genetic outliers, researchers can reconstruct the genetic highways of the past and confirm the existence and extent of ancient interaction spheres. The scale of these networks was continental. Amazonian trade goods such as psychoactive plants and characteristic pottery have been found in high-altitude Andean archaeological sites, while Andean materials like gold and silver have been discovered in the Amazonian lowlands. This transcontinental trade implies not just fleeting contact but sustained relationships between the peoples of the mountains and the peoples of the forest. These interactions are mirrored in the genes. Genetic studies of indigenous groups living at the headwaters of the Amazon, near the Andes, often show a distinct genetic component that links them to Andean populations, a clear signature of long-term bidirectional gene flow across this major ecological boundary. These networks also facilitated the spread of ideas and innovations. The knowledge of how to create terra preta soils, the techniques for cultivating new varieties of manioc, or the complex cosmological and mythological systems that characterize Amazonian spirituality. All of these would have traveled along the same routes as stone axes and feather headdresses. The adoption of a new technology or belief system by a community could make it a more attractive partner for trade or alliance, further integrating it into the network and increasing its genetic connectivity. The social structure of these trade-based societies was often complex and hierarchical, led by powerful chiefs who controlled the production and distribution of prestige goods. This social stratification would also have had genetic consequences. High-status individuals, such as powerful chiefs and their families, may have had greater reproductive success, and their lineages may have spread more widely through the network via strategic marriage alliances. This could lead to a star cluster pattern in the Y chromosome data, where a single dominant male lineage becomes widespread across a large geographic area. The pre-Columbian Amazon, therefore, was not a collection of isolated tribes, but a dynamic, cosmopolitan world connected by commerce and kinship. Its peoples were sophisticated political and economic actors who built a resilient civilization based on reciprocity and exchange. The genetic legacy of these networks is a testament to their success. The DNA of modern Amazonian peoples is a complex mosaic, a rich tapestry woven from countless threads of ancient migration, trade, and alliance, a living record of a time when the rivers and forests were not barriers, but bridges connecting a vibrant, continent-spanning world. Part 6. the cultivated canvas, coevolution with domesticated plants. The genetic history of Amazonian peoples cannot be separated from the genetic history of the plants they cultivated. This was a profound and intimate dance of coevolution, a multi-millennial partnership in which humans and plants domesticated each other, forever altering the biological trajectory of both. The Amazonian forest, often perceived as a natural wilderness, is in many ways a human-created artifact, a vast garden shaped by the selective pressures of its indigenous inhabitants. This process of domestication, which began during the Archaic period and reached its zenith with the rise of agricultural societies, left an indelible signature on the genomes of both the people and their crops, a shared story of mutual adaptation and engineered survival. The cornerstone of Amazonian agriculture was manioc, also known as cassava or yucca, a starchy tuber that was domesticated from a wild progenitor in the southwestern Amazon at least 8,000 years ago. Wild manioc is highly poisonous, containing cyanogenic glycosides that must be meticulously removed before consumption. The invention of a complex, multi-step detoxification process involving scraping, grating, washing, and heating was a major technological breakthrough that unlocked a vast and reliable source of carbohydrates. This cultural innovation had direct genetic consequences. Populations that came to rely heavily on manioc may have evolved enhanced abilities to detoxify the residual cyanide that remained even after processing, a selective pressure that would be written in the genes related to metabolic pathways. At the same time, the plant itself was being transformed. Humans selectively bred manioc for desirable traits: larger tubers, lower toxicity, and faster growth. This artificial selection dramatically altered the plant's genome, making it more productive and more dependent on human cultivation for its survival. The spread of domesticated manioc, carried in the canoes of migrating farmers, can be tracked through the archaeological record and is often correlated with the genetic and linguistic expansions of groups like the Tupi and Arawak. It was a co-dispersal, a journey of a people and their primary plant partner. A similar story unfolded with a host of other plants: the peach palm, cacao, the source of chocolate, pineapple, squash, and peanuts are just a few of the dozens of species that were domesticated or semi-domesticated in the Amazon. Each of these domestications represents a deep understanding of plant biology and genetics, a form of indigenous biotechnology developed over countless generations. The people who tended these plants were not just farmers, they were expert plant breeders. They actively managed the genetic diversity of their crops, maintaining multiple varieties of each species, each adapted to different soil types, rainfall patterns, or culinary uses. This agrodiversity was a key to their resilience, a living insurance policy against disease or climate fluctuations. This deep entanglement with the plant world also shaped the human genome. A diet rich in specific types of carbohydrates, proteins, and plant-based secondary compounds would have created a unique selective environment. Genes involved in nutrient absorption, immune responses to plant-specific pathogens, and the processing of various phytochemicals would have been finely tuned to the local agricultural system. The very landscape became a reflection of this relationship. The distribution of useful plants in forests today is not random. It is a living map of ancient settlements, trails, and fallow gardens. Stands of Brazil nut trees, for example, are now known to be anthropogenic, their high density a clear indicator of a past human presence. These hyperdominant species, which are far more common than would be expected by chance, are a testament to the landscape-level impact of indigenous resource management. The people didn't just clear the forest for their fields, they enriched it, creating a mosaic of managed forest and cultivated plots that was both highly productive and biologically diverse. This process of landscape domestication created what some now call the built forest, and living within this human-modified environment had feedback effects on human genetics. By increasing the abundance and predictability of food resources, agriculture allowed for larger, more sedentary populations. This demographic shift, as noted before, changed the dynamics of gene flow and drift, but it also created a new kind of selective pressure. With a more reliable food source, the selective pressures related to starvation may have been relaxed, while pressures related to diseases of sedentism and crowding would have intensified. The genetic legacy of this coevolutionary partnership is profound. The DNA of Amazonian peoples is a record of a diet and lifestyle built around their domesticated plants. It is a genome that was shaped by the challenges of processing manioc and the benefits of a diet rich in Amazonian fruits and nuts. Conversely, the genomes of those same plants are a record of human ingenuity, a testament to the thousands of years of careful selection and cultivation that transformed them from wild ancestors into the productive crops they are today. This story challenges the very notion of a division between nature and culture. In the Amazon, they are one and the same. The people are a part of the garden, and the garden is a part of the people, a relationship so deep that it is written in their shared DNA. Part 7. Spirits in the Sequence, Cosmology and Genetic Lineage. The worldview of Amazonian peoples is not a secular one. It is a universe saturated with spiritual forces, where the boundaries between humans, animals, plants, and spirits are fluid and permeable. This intricate web of beliefs, which constitutes their cosmology, is not merely an abstract philosophical system. It is a powerful social technology that has had a direct and measurable impact on their genetic history, the rules governing kinship, marriage, and descent which are derived from. These cosmological frameworks have acted as powerful modulators of gene flow, shaping the very structure of the Amazonian gene pool for millennia. Central to many Amazonian cosmologies is the concept of relationality, the idea that identity is defined through one's relationships with others, both human and non-human. Kinship is the primary organizing principle of society, and kinship systems in the Amazon are notoriously complex. They are not just about tracing biological descent, but about creating and maintaining the social alliances necessary for survival. A key feature of many of these systems is the rule of exogamy, the requirement to marry outside of one's own kin group. This seemingly simple rule has profound genetic consequences by forcing individuals to find partners in other communities, often in other villages or even other ethnic groups. Exogamy acts as a powerful engine of genetic exchange, preventing inbreeding and weaving disparate populations into a larger, more resilient genetic network. The specific form of exogamy varies. In some societies, it is based on patrilineal clans where descent is traced through the father's line. In others, it is matrilineal. Many Amazonian societies, particularly in the northwest Amazon, have a system of linguistic exogamy where individuals must marry someone who speaks a different language. This extraordinary system directly promotes gene flow between different ethno-linguistic groups, creating a region of immense cultural and genetic diversity where multilingualism is the norm. The genetic signature of these systems is clear. Y chromosome and mitochondrial DNA lineages become widely distributed across multiple language groups, and the overall genetic differentiation between groups is reduced. Cosmology also informs ideas about ancestry and descent, which are often materialized in rituals, myths, and oral histories. The origin stories of a people frequently describe their emergence from a sacred place or their descent from a mythical ancestor, an animal, a spirit, or a celestial being. These narratives of shared descent create a powerful sense of collective identity that is reinforced through shared ceremonies and communal living. This strong group identity can, in turn, influence genetic patterns. A shared belief in a common ancestor can promote endogamy, marrying within the group at a higher level, for example, within a collection of allied clans who see themselves as one people. This can lead to the formation of distinct genetic clusters that correspond closely to these larger social and cosmological groupings. Shamanism, a cornerstone of Amazonian spirituality, also plays a role in shaping genetic history. Shamans are mediators who travel between the visible world and the spirit world, healing the sick, ensuring hunting success, and maintaining cosmic balance. The power of a shaman is often seen as hereditary, a spiritual essence passed down through specific lineages. These shamanic lineages can become socially and politically powerful, and the families that hold them may engage in strategic marriages to consolidate their spiritual and temporal power. Over generations, this could lead to the disproportionate reproductive success of these specific lineages. a form of social selection that leaves a faint but detectable trace in the patterns of Y chromosome and mtDNA distribution, the world of spirits and non-human. Agents can also influence population dynamics. Beliefs about the spiritual properties of certain places can affect settlement patterns. A sacred mountain, a powerful riverbend, or a forest inhabited by dangerous spirits might be avoided, creating no-go zones that act as barriers to gene flow. Conversely, ceremonial centers, where different groups gather for seasonal rituals, can become hotspots of genetic exchange, drawing people and their DNA from a wide catchment area. Thus, the spiritual landscape and the genetic landscape can become reflections of one another. In this worldview, genes are more than just molecules of inheritance. They are vessels of ancestral essence, the physical manifestation of the kinship lines that stretch back to the mythical first ancestors. When geneticists today draw diagrams of population splits and admixtures, they are, in a sense, mapping the tangible outcomes of these cosmological rules, the decision of who to marry, who to consider kin, and who to regard as other, all of which are guided by the spiritual logic of the cosmos, are the very processes that generate the patterns of genetic variation that scientists study. The resilience of Amazonian peoples, therefore, is not just a matter of biological adaptation, is also a product of their social and spiritual technologies. By creating complex systems of alliance and exchange, guided by a rich cosmological understanding of the world, they built a human ecosystem that was flexible, interconnected, and enduring. The spirits, acting through the rules of kinship and ritual, have been silent but powerful architects of the Amazonian genome. Part 8. The Great Silence. Pathogens of the Columbian Exchange. The year 1492 marks a rupture in the timeline of the Americas, a moment that severed the ancient, isolated evolutionary trajectory of its peoples and plunged them into a new and terrifying biological world. The arrival of Europeans initiated the Columbian Exchange, a vast intercontinental transfer of plants, animals, technologies, and ideas. But the most consequential and devastating part of this exchange was the invisible cargo carried by the newcomers, a host of infectious diseases for which the indigenous peoples of the Americas, including those in the vast Amazon basin, had no acquired or evolutionary immunity. The result was a demographic catastrophe of unimaginable proportions, a great dying that swept through the continent, leaving a profound and permanent scar on the genetic landscape of its survivors. For at least 15,000 years, the ancestors of Amazonian peoples had lived in virtual epidemiological isolation. Their journey across the Bering Land Bridge had acted as a disease filter, as small, mobile hunter-gatherer bands could not sustain crowd epidemic diseases, and the frigid climate would have killed off many pathogens. In the Americas, they encountered a different suite of pathogens, but they were spared the great pestilences that had shaped old world history. Smallpox, measles, influenza, bubonic plague, typhus, and diphtheria. These diseases, which had co-evolved with dense, urban, agricultural populations in Europe, Asia, and Africa, where they had become endemic childhood diseases, were completely alien to the American immune system. When they were introduced, they spread not as epidemics, but as relentless, explosive pandemics through a virgin soil population. The impact was apocalyptic. Smallpox, often the first and most lethal of the imported killers, could have a mortality rate of 50 to 90% in a non-immune population. It traveled faster than the Europeans themselves, passed from one indigenous community to another along the very trade networks that had been the arteries of pre-Columbian life. A single infected individual arriving in a densely populated Amazonian town could trigger a chain reaction that would wipe out entire communities in a matter of weeks. The genetic consequences of this cataclysm were immense. The most immediate effect was a massive and severe population bottleneck. It is estimated that up to 95% of the indigenous population of the Americas perished in the centuries following contact. In the Amazon, where large sedentary populations had flourished along the major rivers, the die-off was particularly severe. This drastic reduction in population size led to a catastrophic loss of genetic diversity. countless unique mitochondrial and Y chromosome lineages, representing thousands of years of evolutionary history, were extinguished forever. The genetic variability that is the raw material for adaptation and resilience was simply wiped from the slate. This bottleneck is clearly visible in the DNA of modern indigenous Amazonians, who, despite their cultural diversity, often exhibit surprisingly low overall genetic diversity compared to populations in other parts of the world. Beyond the sheer loss of diversity, the pandemic waves acted as an incredibly powerful, if brutal, selective event. In the midst of the carnage, some individuals did survive. Their survival was likely due to a combination of factors, including pure chance, but it is almost certain that genetic variation in the immune system played a critical role. Individuals who happened to possess rare genetic variants in their human leukocyte antigen, HLA genes, or other immune response pathways that conferred even a slight advantage against the novel pathogens were more likely to survive, reproduce, and pass on those life-saving alleles to the next generation. This process, known as a selective sweep, would have rapidly increased the frequency of these protective variants in the surviving populations. Modern genetic studies have identified several such signatures of selection in the genomes of indigenous Americans, centered on genes involved in immunity. These are the genetic echoes of the Great Plagues, a testament to the ancestors who survived against impossible odds. The social and cultural collapse that accompanied the demographic collapse further reshaped the genetic landscape. Complex societies disintegrated. Vast, engineered landscapes with their terra preta soils were abandoned and reclaimed by the forest. Survivors from different ethnic groups, their own communities shattered, were forced to band together in new, multi-ethnic settlements, often in remote, inaccessible locations deep in the forest. This process of ethnogenesis, the creation of new peoples from the remnants of the old, led to novel patterns of genetic admixture. The genetic boundaries that had once separated different polities and language groups were dissolved and redrawn, creating a new, more homogenized, but also more fragmented genetic reality. The Amazon that Europeans discovered was in many ways a widow, a landscape haunted by a recent and profound trauma, its surviving inhabitants living in the shadow of a lost world. The Great Silence, as some have called it, the eerie emptiness of lands that were once teeming with people, was a direct result of this microbial holocaust. The genes of the survivors tell this story of loss and survival, of a biological bottleneck so severe it nearly extinguished them, but also of the remarkable resilience that allowed them to endure, carrying forward a precious, though diminished, fragment of the immense genetic diversity that had existed before the deluge. Part 9. Islands of Humanity. Post-contact genetic sanctuaries. In the wake of the Great Dying, as old world pathogens continued to sweep through the Amazon in devastating waves, the fundamental patterns of human settlement and genetic structure were irrevocably altered. The once thriving heartlands along the major riverine arteries, which had been home to large, interconnected, and genetically diverse agricultural societies, became death traps. These rivers, the former highways of commerce and gene flow, now served as vectors for disease, facilitating the rapid spread of epidemics deep into the continental interior. For the indigenous peoples who survived the initial onslaught, the only viable strategy for survival was retreat. This period, stretching from the 16th to the 19th centuries, was characterized by a profound demographic and geographic reorganization, a turning inward that transformed the Amazonian genetic landscape into an archipelago of isolated human sanctuaries. The survivors abandoned the exposed and dangerous riverbanks and fled into the remote interfluvial highlands, the areas of dense, elevated forests situated between the major river systems. These regions, which had previously been sparsely populated by small, specialized forager groups, now became refugia, havens from the chaos and disease that consumed the lowlands. In these isolated pockets, fragmented communities of survivors, often composed of people from multiple, shattered ethnic groups, began the arduous process of rebuilding their societies and their gene pools. This flight into the deep forest had profound genetic consequences. The primary effect was intense and prolonged genetic isolation. The former web of long-distance trade and alliance was severed. Contact with the outside world, now a source of mortal danger, was actively avoided. This isolation, coupled with the small size of these new refugee populations, created the perfect conditions for genetic drift to act with immense power over generations. The random fixation and loss of alleles caused these small, isolated groups to diverge rapidly from one another. A rare genetic variant present in one founding group could, by sheer chance, become common in their descendants, while being completely absent in a neighboring refugial population just a few dozen miles away. This process led to a dramatic increase in interpopulation genetic differentiation. The Amazonian gene pool became highly structured, with each island of humanity developing a unique and distinct genetic signature. The social organization within these refugia also contributed to this pattern of differentiation. Faced with existential threats, communities became more insular and ethnocentric. Rules of endogamy, marrying within the group, were likely strengthened to maintain social cohesion and cultural identity in the face of collapse. This practice further restricted gene flow between groups, amplifying the effects of genetic drift and accelerating their genetic divergence. The result was the formation of the tribal mosaic that is often seen as the timeless, ancestral state of the Amazon, but is in fact a relatively recent, post-contact adaptation. The fierce autonomy and distinct cultural and genetic identity of many modern Amazonian groups is not a relic of the pre-Columbian past, but a product of this history of fragmentation and isolation, a survival strategy forged in the crucible of the colonial apocalypse. Yet, these refugia were not just places of genetic loss and divergence, they were also crucibles of ethnogenesis and adaptation. As survivors from different linguistic and cultural backgrounds were thrown together, they created new, syncretic cultures and new, admixed genetic identities. Oral histories often recount how a new community was founded by a handful of families who escape a massacre or a plague, bringing with them their own unique traditions and, of course, their own unique DNA. The resulting populations are a complex genetic tapestry woven from the threads of these different ancestral groups. Furthermore, these isolated populations continued to adapt to their specific local environments. The interfluvial forests presented different ecological challenges and opportunities than the riverine floodplains. The diet, the local pathogens, and the demands of subsistence were different. And so natural selection continued its slow but persistent work, fine-tuning the genomes of these populations to their new homes. A gene variant that was advantageous in the highlands might be different from one that was advantageous in a swampy lowland area, leading to subtle patterns of local genetic adaptation on top of the broader patterns of drift. Reading the genetic map of the modern Amazon is therefore like reading a palimpsest. Beneath the surface of the current distribution of peoples, with their distinct languages and genetic profiles, lie the fainter traces of the older, pre-contact world, the signatures of the great linguistic expansions and the interconnected trade networks. But superimposed on top of this is the strong, clear script of the post-contact era, the evidence of a massive population bottleneck, the radical restructuring of the population into small, isolated groups, and the powerful differentiating effects of genetic drift. The Amazonian archipelago of human diversity is a direct legacy of this period, a testament to the ability of its peoples to find sanctuary in the deepest recesses of the forest and to preserve their cultural and genetic heritage against overwhelming odds, transforming the genetic landscape into a mosaic of resilient, isolated islands of humanity. Part 10. The Ghost Races, Forging Identity Amidst Collapse. The centuries following the European invasion were a time of unparalleled chaos, a period when the very social and ethnic fabric of the Amazon was torn asunder, as indigenous societies collapsed under the combined weight of disease, warfare, and enslavement. The carefully constructed categories of identity that had structured the pre-Columbian world, the intricate systems of kinship, language, and political affiliation began to dissolve. From the ashes of this destruction, new peoples were born. This process of ethnogenesis, the creation of new ethnic and social identities, was a critical survival strategy, but it has created a complex and often confusing picture for modern geneticists and anthropologists trying to map the connections between contemporary groups and their pre-contact ancestors. The DNA of many modern Amazonian peoples tells a story of profound disruption and novel admixture, the story of the forging of ghost races from the remnants of a lost world. A primary driver of this process was the catastrophic depopulation itself. With mortality rates as high as 95%, entire ethnic groups, languages, and cultures vanished from the face of the earth, leaving behind only a handful of survivors. These survivors, stripped of their social context, were forced to seek refuge wherever they could find it. A family whose village was wiped out by a smallpox epidemic might travel for weeks through the forest until they encountered another small group of survivors, themselves from a different, though perhaps related, people. Together, these fragments would coalesce to form a new community. In this new settlement, old animosities might be forgotten, and former distinctions of language and custom would begin to blur. They would need a new name for themselves, a new collective identity to make sense of their shattered world. Genetically, this process resulted in the creation of admixed populations whose DNA contained lineages from multiple distinct pre-contact sources. A single modern indigenous group might carry mitochondrial DNA haplogroups characteristic of a Tupian group, Y chromosome lineages from a Gus-speaking people, and autosomal DNA that shows influences from a now-extinct Arawakan polity. Their genome is a mosaic, a testament to the desperate alliances and forced mergers that characterize this chaotic era. This makes drawing a straight line from a modern tribe to a single pre-Columbian ancestor impossible. The lines of descent are tangled and broken, a web rather than a tree. The mission system, established by Jesuit, Franciscan, and other Catholic orders, was another powerful engine of ethnogenesis. The missionaries' goal was to reduce the Indians from their scattered autonomous settlements into large, centralized mission towns, aldeas, or reductions. In these towns, people from dozens of different ethnic and linguistic backgrounds were brought together, often against their will. While the missions offered a degree of protection from slavers, they were also epidemiological death traps where diseases spread rapidly. Within the mission walls, a systematic process of cultural homogenization was enforced. Indigenous languages were suppressed in favor of a lingua franca, like Tupi-esque lingua geral in Brazil, and traditional social structures were replaced with a new Christian order. This forced cohabitation and cultural stripping led to a massive amount of interethnic marriage and admixture. People whose ancestors had been enemies for generations were now neighbors, their children playing together and eventually marrying, mingling their once distinct gene pools. When the mission systems eventually collapsed, the inhabitants dispersed, but they did not return to their ancestral identities. They left as a new people, the Mission Indians. With a shared history of survival and a highly admixed genetic heritage, the Mura people of the central Amazon are a powerful example of this process. In the 18th century, they were a powerful, non-agricultural riverine people who fiercely resisted Portuguese expansion. After being militarily defeated, the survivors were settled and assimilated. Today, the Mura are genetically one of the most admixed indigenous groups in the Amazon. Their DNA, a complex blend of their original ancestry with that of African slaves, European settlers, and other indigenous peoples with whom they were thrown into contact. Their story is not unique. Many of the groups known today as Caboclos or Riberanos, the river-dwelling, mixed ancestry populations of the Amazon, have their origins in this period of intense social and genetic amalgamation. They are the living descendants of these ghost races. Peoples whose original ethnic labels have been lost to history, but whose genetic legacy persists in the vibrant mix of their DNA. Understanding this history is crucial for interpreting modern genetic data. It warns against a simplistic, essentialist view of indigenous identity, the idea that each tribe is a pure, unchanging biological and cultural unit. The reality is far more dynamic. The DNA of Amazonian peoples is a story of resilience through recombination, of survival through the forging of new identities from fragments of the old, a testament to the creative human capacity to build new worlds even as the old one is burning down around them. Part 11. Chains of Extraction: The Genetic Scars of Forced Labor. Following the initial demographic collapse caused by disease, the surviving indigenous populations of the Amazon were subjected to a new and equally destructive force: a brutal system of economic extraction built on forced labor. From the 16th century onward, the basin's rich natural resources, precious metals, exotic woods, and later rubber, were plundered to fuel the economies of colonial and national powers. This centuries-long campaign of exploitation inflicted deep and lasting wounds on the genetic landscape of the Amazon, not through microbes, but through the systematic enslavement, displacement, and extermination of its peoples, leaving a legacy of demographic disruption and sex-biased genetic admixture that is still clearly visible today. The earliest phase of this exploitation was driven by the slave trade. The Portuguese in Brazil and the Spanish in the western Amazonian territories organized expeditions, known as banderas or entradas, deep into the interior to capture indigenous people and force them to work on coastal plantations and in mines. These slave raids were incredibly violent and destructive. Entire communities were wiped out, and those who were not killed were shackled and marched hundreds of miles away from their ancestral lands, never to return. This process had a devastating and gendered impact on the indigenous gene pool. The raiders primarily sought able-bodied young men for heavy labor, while women and children were also captured for domestic servitude. In the source populations, the removal of a significant portion of the male population would have skewed the sex ratio, disrupted kinship systems, and reduced overall genetic diversity. In the destination areas, such as the coastal plantations, the captured indigenous people were thrown into a melting pot with enslaved Africans and European colonists. Here, a process of intense and often violent admixture occurred. Indigenous women were systematically subjected to sexual violence by European colonists and slave owners, resulting in the introduction of European Y chromosomes into a Native American mitochondrial DNA background. This pattern of sex-biased admixture, where the paternal ancestry is overwhelmingly European and the maternal ancestry is overwhelmingly indigenous or African, is a stark genetic signature of the power dynamics of the colonial world, a record of sexual exploitation written in the genome of modern Brazilian and other Amazonian country populations. The so-called rubber boom of the late 19th and early 20th centuries represents the apex of this extractive brutality. The invention of vulcanized rubber created an insatiable global demand, and the Amazon rainforest was the world's sole source of the latex-producing Hevea brasiliensis tree. A legion of ruthless rubber barons descended upon the region, enslaving local indigenous populations to tap the rubber trees. The system they created was one of pure terror. Indigenous communities were conquered, and their people were forced into debt peonage, given impossible quotas for rubber collection, and subjected to torture, mutilation, and mass murder if they failed to meet them. This period, known as the Devil's Paradise, led to second great dying in many parts of the Amazon, particularly in the Putumayo region of Colombia and Peru. The genetic consequences were horrific. Entire ethnic groups were driven to extinction. The demographic pyramid was inverted, with the loss of a huge number of young men and women of reproductive age. The constant movement of labor gangs, marching from one rubber tapping area to another, further disrupted settlement patterns and kinship structures. It also facilitated a grim form of gene flow, as captured people from different regions were forced to live and work together, and their captors fathered children with local women. The genetic diversity of a region was massively reduced, and the survivors were left with a legacy of trauma and demographic instability. Even after the collapse of the rubber boom, this extractive logic continued in other forms: logging, gold mining, and cattle ranching. Each of these industries brought new waves of outsiders into indigenous territories, leading to further violence, displacement, and disease. The construction of roads, like the Trans-Amazonian Highway in the 1970s, cut through previously isolated territories, acting as new vectors for disease and colonization. and triggering a new round of contact and demographic collapse for previously uncontacted peoples. The genetic scars of this long history of exploitation are multifaceted. They include the drastically reduced genetic diversity caused by repeated population crashes, the specific geographic patterns of European and African admixture that trace the routes of slavery and economic penetration, and the signatures of sex-biased gene flow that speak to a history of sexual violence. The DNA of the Amazon's peoples is therefore not just a record of their deep ancestral history. It is also a testament to their survival in the face of a relentless, centuries-long assault on their bodies, their lands, and their very existence, a genetic archive of the chains of extraction. Part 12. Echoes in Living Memory. Oral Histories Meet Genetic Science. For generations, the history of the Amazon's indigenous peoples was a story told by outsiders, by conquistadors, missionaries, explorers, and anthropologists. Their own rich, detailed accounts of their past, preserved in the intricate tapestry of oral tradition, were often dismissed as myth or folklore. These narratives, passed down from elder to child through spoken word, song, and ritual, speak of great migrations, of cataclysmic floods and fires, of ancient alliances and epic wars, of the founding of new communities from the ashes of old ones. In recent years, a powerful new dialogue has begun, one that places these indigenous histories alongside the findings of modern population genetics, revealing a stunning degree of corroboration and offering a richer, more nuanced understanding of the Amazonian past. This convergence of living memory and molecular data is rewriting the history of the region, not from an external perspective, but from the inside out. A compelling example of this synergy comes from a study of the Tupian expansion. The oral histories of many Tupi-speaking groups, from the Guarani in the south to the Tupinamba on the coast, tell of a relentless search for a mythical land without evil, a terrestrial paradise that drove their ancestors to undertake long and arduous migrations away from their ancestral homeland. For decades, this was interpreted metaphorically. Genetic research, however, has provided a biological correlate to this foundational myth. By analyzing the Y chromosome and mitochondrial DNA of numerous Tupi-speaking groups and comparing it with that of other Amazonian peoples, geneticists have been able to reconstruct the geographic origin and migratory pathways of the Tupian expansion. The genetic data points to a homeland in what is now Rondonia, in the southwestern Amazon, and traces a pattern of dispersal that closely matches the routes and chronologies preserved in the oral traditions. The myth, it turns out, was a form of historical memory, a stylized but factually grounded account of one of the most significant demographic events in the continent's prehistory. Similarly, the genetic signatures of the Great Dying and the subsequent retreat into isolated refugia find powerful echoes in indigenous narratives. Many Amazonian cosmologies are punctuated by a deep sense of a lost world, a past golden age of great cities and powerful chiefs that was brought to a catastrophic end. Elders tell stories of a time when their people were as numerous as ants before a great sickness, a wind of death, swept through the land and silenced the world. They speak of their ancestors fleeing into the deep forest to escape the chaos, of small bands of survivors banding together to create new communities. This is precisely the scenario that geneticists have reconstructed from the DNA. A severe population bottleneck, the collapse of large, ancient populations, and the subsequent founding of new, smaller groups from a limited number of survivors, leading to increased genetic drift and differentiation. The oral histories provide the lived experience, the human trauma and agency behind the abstract patterns of genetic data. The convergence works in the other direction as well. Genetics can sometimes help to clarify or resolve ambiguities within the oral traditions. For instance, two groups who speak different languages and consider themselves distinct peoples might have an oral history that hints at a shared ancient ancestry. Genetic analysis can test this hypothesis directly. If they are found to share a recent common ancestor, evidenced by long, identical segments of DNA, it can confirm the historical connection, revealing that their cultural and linguistic divergence is more recent than their shared biological origin. This has profound implications for indigenous peoples themselves, as it can provide scientific validation for their own understanding of their history and relationships with neighboring groups, sometimes bolstering land claims or political alliances. This collaborative approach, which brings indigenous elders and leaders into the research process as equal partners, is transforming the ethics and practice of anthropological genetics. Instead of being passive subjects of study, indigenous communities are becoming active participants in the scientific exploration of their own past. They are helping to frame the research questions, to interpret the genetic findings in the light of their own knowledge systems, and to ensure that the research is conducted in a way that is respectful of their cultural values and sovereignty. This dialogue between the ancient library of oral tradition and the modern library of the human genome is creating a history that is more than the sum of its parts. It is a history that is at once scientifically rigorous and deeply human, one that honors the voices and memories of the people whose story it tells. The echoes of the past, preserved for centuries in the spoken word, are now being heard and amplified in the language of DNA, a powerful testament to the resilience of memory and the enduring truth carried in the blood. Part 13. The Digital Teepee, Technology's Role in Modern Research. The exploration of the Amazon's genetic history has been propelled by a series of technological revolutions that have allowed scientists to read the book of life with ever-increasing speed and precision. Just a few decades ago, the idea of sequencing the entire genome of a single individual was the stuff of science fiction, a multi-billion-dollar, decade-long endeavor. Today, thanks to the advent of high-throughput sequencing and sophisticated computational biology, researchers can analyze the DNA of thousands of individuals from remote Amazonian communities, unlocking a treasure trove of information about their deep past, their adaptations to the forest environment, and their relationships with other populations. This new era of genomic research, facilitated by what could be called a digital teepee of portable labs and powerful computers, is painting a picture of Amazonian history that is unprecedented in its detail and scope. The workhorse of modern population genetics is next-generation sequencing, NGS. Unlike older methods that could only look at a few genetic markers at a time, like mitochondrial DNA or a handful of Y chromosome markers, NGS technologies can rapidly sequence millions or billions of DNA fragments simultaneously. This allows researchers to move from looking at a few sentences to reading the entire book of the genome. By comparing the complete genomes of different individuals and populations, they can identify millions of single nucleotide polymorphisms, SNPs, the tiny variations in the DNA code that make each person unique. The patterns of these SNPs, how they are shared where they differ, are the raw material for reconstructing history. They allow scientists to build highly detailed family trees of human populations, to estimate with remarkable accuracy when different groups diverge from one another, and to detect the subtle signatures of events like population bottlenecks, migrations, and admixtures. Another powerful tool in the geneticist's arsenal is the analysis of ancient DNA ADNA. The humid, acidic environment of the Amazon is notoriously poor for preserving organic remains, but in recent years, new techniques have allowed scientists to successfully extract and sequence DNA from ancient human bones and teeth found in archaeological sites, some dating back thousands of years. This is a game changer. Instead of having to infer the past solely from the DNA of living people, scientists can now directly observe the genetic makeup of past populations. They can see what the gene pool of the terra preta makers looked like before European contact, or directly test for the presence of the mysterious population Y signal in the Pleistocene-era inhabitants of the basin. Ancient DNA provides a direct window into the past, allowing researchers to calibrate the molecular clock and to witness evolutionary processes like the selection for immune genes during the great epidemics as they happened. This deluge of genomic data from both modern and ancient individuals requires immense computational power to analyze. The field of bioinformatics has emerged to meet this challenge, developing sophisticated statistical algorithms and machine learning techniques to sift through the billions of data points and extract meaningful historical signals. Researchers can now use powerful computer models to simulate different historical scenarios, a single migration versus multiple waves, for example, and then see which model best fits the observed genetic data. They can create detailed geographic maps of gene flow, identifying the corridors and barriers that shaped human movement over millennia. They can even identify segments of the genome that have been subject to positive natural selection, pointing to the specific genes that helped Amazonian peoples adapt to their unique environment. The technology is also becoming more portable, while large-scale sequencing still happens in major labs. New, pocket-sized sequencing devices, coupled with solar power and satellite internet, are making it possible to conduct genetic analysis in the field, even in the most remote villages. This allows for a more collaborative research model, where preliminary results can be shared and discussed with the community in real time, making the process more transparent and empowering for the indigenous participants. This digital teepee, a convergence of genomics, archaeology, and computational science, is allowing us to ask and answer questions about the Amazonian past that were previously unanswerable. It is revealing a history of deep antiquity, complex migrations, and profound resilience that was invisible to previous generations of researchers. It is a technological revolution that, when guided by an ethical framework of collaboration and respect, has the power to decolonize the scientific narrative and restore the deep and complex history of the Amazon to its rightful owners. Part 14. The Genome as Homeland, Data Sovereignty, and Indigenous Rights. The explosion of genomic research in the Amazon, while offering unprecedented insights into the region's deep history, has also raised profound ethical and political questions that strike at the very heart of the relationship between science and indigenous peoples. For centuries, indigenous communities have been the subjects of outside study, their bodies artifacts, and knowledge extracted and analyzed with little to no consultation, consent, or benefit. Genetic material, the most intimate and fundamental component of a person's identity, became the latest resource to be extracted, a phenomenon often described as bioprospecting or biopiracy. In response to this history of exploitation, a powerful global movement for indigenous data sovereignty has emerged, demanding that indigenous peoples have the right to control their own biological and cultural information. This principle, encapsulated in the concept of the genome as homeland, reframes genetic data not as a universal human resource to be freely mined by science, but as a collective ancestral inheritance that belongs to the community from which it came. At the core of this movement is the principle of free, prior, and informed consent (FPIC). This goes far beyond the simple signing of a consent form. It requires that communities are fully and transparently informed about all aspects of the research, its goals, methods, potential risks, and benefits, in a culturally appropriate manner and in their own language, long before any research begins. It also means they have the absolute right to say no, to opt out of the research at any stage, or to place specific restrictions on how their genetic data can be used, stored, and shared. This fundamentally shifts the power dynamic from an extractive model to one of genuine partnership, where the community is an equal collaborator in the scientific process. A key challenge is the issue of data ownership and governance. When a blood or saliva sample is taken from an indigenous person and sequenced in a lab thousands of miles away, who owns the resulting data? Who decides who gets to access it, and for what purpose? Historically, the answer has been the researcher or their institution. The indigenous data sovereignty movement challenges this assumption, arguing that the community should retain governance rights over their own genomic data. This has led to the development of new models for data sharing, such as community-controlled biorepositories and data enclaves, where access to the data is managed by a council of community representatives according to collectively defined rules. It also involves creating legally binding benefit-sharing agreements that ensure the community receives tangible benefits from the research, which could range from financial compensation and co-authorship on scientific publications to investment in local health and education projects. The stakes are incredibly high. Unregulated genetic research can cause real harm. It can reveal information about paternity or ancestry that contradicts a community's oral traditions or social structure, causing internal conflict. It can lead to the stigmatization of a group if they are found to have a genetic predisposition to a certain disease. Furthermore, there is the risk of commercial exploitation. A unique genetic variant found in an indigenous population could be patented by a pharmaceutical company for drug development, with no benefit returning to the community whose ancestral lineage produced that variant. By asserting their data sovereignty, indigenous peoples are not rejecting science. On the contrary, many communities are eager to use genomic technologies to explore their own history, to understand their health, and to strengthen their identity. What they are rejecting is a colonial model of science in which they are treated as passive objects of study. They are demanding a new paradigm, one based on respect, reciprocity, and self-determination. This has led to the rise of a new generation of indigenous scientists, researchers who come from these communities, and are combining rigorous scientific training with their own cultural knowledge to lead research projects that are accountable to their own people. They are building capacity within their communities to manage their own genetic resources and to conduct their own research. The struggle for genetic sovereignty in the Amazon is the modern frontier of the long struggle for indigenous rights. It is the fight for control over their lands, their bodies, and their stories. It recognizes that the genome is not just a sequence of letters, but a sacred archive, a landscape of ancestry, a homeland encoded in DNA. Securing the right to govern this homeland is a crucial step in ensuring that the future of the Amazon's peoples is not one of continued extraction, but of self-determined resilience and cultural revitalization. Part 15. Reweaving the Ancestral Fabric, a Future Written in DNA. The story of the Amazon's unseen DNA is, in its final telling, a story of hope. It is a narrative that, while etched with the deep scars of cataclysm and exploitation, is ultimately defined by an astonishing and enduring resilience. As we move into the 21st century, the same genomic technologies that have revealed this complex past are now being harnessed by indigenous peoples themselves as powerful tools for cultural revitalization and self-determination, far from being a final chapter that closes the book on their history. The mapping of the Amazonian genome is providing a new set of threads, spun from the double helix, with which to reweave the ancestral fabric and to chart a course for a self-determined future. For many indigenous communities, the scientific validation of their deep ancestral connection to their lands is a powerful weapon in the ongoing struggle for territorial rights. In legal battles against governments and corporations seeking to exploit their territories for mining, logging, or agribusiness, genetic evidence can be a powerful complement to archaeological findings and oral histories, demonstrating a unique and ancient genetic lineage that is tied to a specific geographic region can bolster a community's claim as the original and continuous inhabitants of that land, strengthening their case under national and international law. The genome becomes a legal document, a testament of ownership written not in ink, but in the base pairs of their DNA, providing a form of evidence that is often compelling to outside legal and political systems. Beyond the courtroom, genomics is also playing a role in the revitalization of cultural identity for communities whose histories have been fragmented by colonization, whose oral traditions have been suppressed, and whose links to their ancestors have been severed. Genetic ancestry testing can provide a tangible, biological connection to the past, learning that their DNA carries the signatures of the great Arawak migrations or connects them to the ancient makers of terra preta can foster a renewed sense of pride and a deeper understanding of their place in the long saga of Amazonian history. It can help to reconnect communities that were separated by colonial violence, revealing shared genetic histories between groups who now speak different languages and live hundreds of miles apart, opening the door for new political alliances and cultural exchanges. This process is not without its complexities, as a biological definition of identity can sometimes clash with a community's own social and cultural definitions of kinship and belonging, but when driven by the community itself, it can be a powerful tool for rebuilding a fractured sense of self. In the realm of health, the field of indigenous genomics holds the promise of a more equitable and effective form of medicine. For too long, medical research and treatment have been based on a one-size-fits-all model, predominantly based on populations of European descent. But the unique genetic history of Amazonian peoples, their deep time in isolation, their adaptation to a tropical environment, and their experience with severe population bottlenecks has resulted in a distinct genetic architecture. This means they may have different susceptibilities to certain diseases and may respond differently to certain medications. By studying their own genomes, indigenous communities, in partnership with ethical researchers, can develop personalized medicine approaches that are tailored to their specific genetic background. This can lead to better prevention and treatment strategies for prevalent health issues, from infectious diseases to metabolic disorders like diabetes, empowering them to take control of their own health and well-being. Perhaps most profoundly, the journey into the Amazonian genome is fostering a new generation of indigenous leaders and scientists. Young people from remote villages are now pursuing careers in genetics, bioinformatics, and anthropology, bringing their unique cultural perspectives to these fields. They are creating a new, decolonized science, one that integrates the rigor of the scientific method with the wisdom of traditional ecological knowledge. They are building the infrastructure, the community-controlled labs, and data centers that will allow them to be the primary stewards and interpreters of their own genetic heritage. They are the new guardians of the living library, ensuring that the story told by their DNA is their own. The future of the Amazon's indigenous peoples will be written in many languages, in their revitalized native tongues, in the language of international law, and in the digital code of their own sequenced genomes. The unseen DNA, once a passive record of the past, is becoming an active blueprint for the future, a source of knowledge, power, and pride that will fuel their continued resilience for generations to come.
The story of the Amazon is not over. It continues in the courage of its people and the resilience encoded in their DNA. By understanding their past, we honor their present and support their future. Thank you for watching and join us next time for more hidden histories.